I decided to talk about potentially two different tales of why in certain cases something happened, in certain cases it's much more difficult. And for that, I decided to focus on two applications of CEST, endogenous CEST and effectively GLUCOCEST. So this is my declaration of financial interest. So at the beginning, there was a new world, a world of options, CEST. So to understand why it was like that, well, this is the signal that we are usually looking at. That we were looking at with MRI. It's a water signal. It's big, and it's so great that you can actually, in preclinical imaging, for example, make beautiful images at very, very high resolution, right? Now, if you're interested in the chemistry that's happening into these tissues, well, you need to do something. The first thing is, you need to kill the water signal, and then if you want to measure anything, you see that you need to amplify this scale by 3 orders of magnitude so that you can actually detect something. And then, of course, your resolution becomes much, much broader. Like, rather than these beautiful high-resolution images, you have these big voxels. So can we have the best of both worlds? And that's where CEST, or chemical exchange saturation transfer, comes. So I'm going to focus only on application of cancer, and the interesting thing about cancer is, first of all, it has some very big differences in the way it metabolized the glucose. While in differentiated tissue, as long as you have oxygen, you produce 36 mole of ATP per molecule of glucose by getting the glucose, the pyruvate, to actually be transferred into the CTA, the citrus cycle. But if you're in hypoxia, there is a way to try to get a little bit of energy by actually transforming the pyruvate into lactate. Now, tumor, for weird reason, have decided to go that particular way primarily as their way of providing energy, and therefore this so-called aerobic glycolysis, or the Warburg effect, makes it a very energy inefficient way of producing energy. From a molecule of glucose, you only get 4 molecules of ATP. So for this, it's going to change a lot of things. It's going to change the way the metabolism, the way the cytosol works, and everything beyond. So glucose detection has been going on for a long time, and this is an FDG PET image showing here tumors in the liver. And on the other side, because the metabolism has been changed so much, there is a large amount of amide protons here that could potentially be detected, and that's the endogenous method that was referred to already in the plenary lecture and Lauterbur lecture by Professor Van Zyl. So when I was at Peter's lab, Ginny and Zhao had started working on this on this method, and this was one of the very early work on trying to detect a new contrast in brain tumors. And this new contrast seems to be actually interesting, as it could actually differentiate the tumor from the surrounding tissue. Not only it could differentiate the tumor from surrounding tissue, but in cases where you have radiation necrosis or that could be mistaken for tumor progression, because in case of radiation necrosis, there is no exchange. There's no actually really molecules that can actually exchange that easily. You will have basically a dark contrast, while in the case of in the case of a tumor progression, you will have a bright contrast. So it's pretty much the dream, right? If you want to have a method that separates between two conditions, one is black, the other is white. So actually this, in the meantime, has been already applied, but that was very interesting work at the time. As Silvio mentioned, a couple of years later, we were trying to see whether we could actually replace FDG using simple glucose, because, well, it's cheaper, it's more available, and to be fair, I've never really liked radiations. So then we had these two different animal models of of colorectal carcinoma, and these are just like this traditional flank model in a nude mice, and you see the tumor here and the surrounding tissue, and if you're measuring the signal at baseline and after injection of glucose, the difference between the two lights up the tumor and not the surrounding tissue. In this case, it lit up a little bit the surrounding tissue, but that was actually a direct inflammatory response just below this very rather large 1 centimeter tumor in this animal. So it seemed to work. We wanted to compare with FDG. So for those of you who are not biochemists in the room, this is a molecule of glucose and this is a molecule of FDG. So it's actually relatively similar. They behave very differently from in a metabolic perspective. FDG gets trapped. So if you were to actually use 2-deoxyglucose, you could use it. However, there will be slightly toxic in the concentration that we need in MRI. But FDG inject picomolar or less concentration of the agent, and therefore you can actually use whatever kind of the most toxic poison on earth, and you could probably detect where it goes with FDG without any problem. So these were also presented by Sylvia already, so we could actually show that actually we had a differentiation between these two colorectal models. One had a high metabolism, the other a low metabolism, and you could see a high signal in the FDG and a high signal in the GLUCOCEST. On the other side of the pond, at Hopkins, people were doing the same thing. Not surprising. I mean, I'm coming from that group. I had this idea, and they were working on the same idea. So completely independently. But in their case, they didn't actually, and that was the first hint that maybe that would have been something a bit more complicated than just simply the endogenous CEST. That actually, in their case, they couldn't actually see a good correlation between FDG and GLUCOCEST. So we decided after all this time to go into clinical practice. So we got this GLINT grant, and we were trying to optimize the signal. So in the first place, let's move back to this endogenous CEST. In the early days, this is from the same paper that Sylvia already mentioned. So this was just another case, and you can see that if you can see the tumor in this case, well, you have still at these early days you had a lot of artifacts here. This very prominent signal here is actually just due to the fact that you have a shift in the B0 fields that was not completely corrected for at that time, and in which case it appears as an abnormal signal, while there is nothing here abnormal in this patient. So it needed some optimization. This is just one of the most potentially difficult field, and this optimization is quite important, as if you're looking at here, these different colors in this case refer to the different main magnetic field. Because the exchange is dependent on the separation of the frequency from the exchangeable group to water, it will scale. This effect will scale differently at different magnetic fields because this frequency difference will be different. And therefore, while for AMIDE groups, you basically have a very good labeling efficiency no matter what you do, pretty much at any kind of field strength, except if you're going to very, very high amplitude. But nobody would actually try to put this type of radiation in a patient in a clinical scanner, because that would be actually over 2 to 3 seconds. That will surpass any kind of regular problem. So yeah. Any kind of regulation. So anyway, for our amine, and the same for hydroxyl group, the problem you will have is actually you will, with a lower and lower field strength, if you're going from the very high field that you have here, like a B0 at 15 Tesla or 14.7 Tesla, you have a very high and nice labeling efficiency. At 3T, you start having a much lower labeling efficiency. That means your signal is going to be much lower. And this is just due to the fact that these are actually exchanging too fast at 3T for them to be detected properly. So my good friend here, Moritz, did a lot of with his students did a lot of optimization based on this consideration, and he also actually showed that actually when you're doing things in clinical practice, you need to take care of something more problematic, like your patient is alive. It will move. And these small motions, when you detect a very, very tiny signal, can be very problematic. This is a very well-known problem. Everybody who has ever done fMRI knows about it. However, it is more complicated in there because actually a single scan is made of multiple acquisition. So we managed to do a little bit of all of this. We optimize everything. And here some of the early results of actually measuring this different patients, in this case, in 3 different hospitals at UCL, at the Max Planck, and in Tel Aviv University. And you could see that actually you can reliably and over time measure the same things pretty much with a relatively small difference in a healthy volunteer. And doesn't tell you that you're going to have a very high reproducibility in patients, but it's a first step. So let's move to the clinical reality, or talk about the clinical readiness level. You know, you have this TRL, this is a CRL. Are we there? Well, actually, in our hospital, and this is a slide from one of our neuroradiologists, Dr. Bistas, is using CEST pretty much on every patient with glioma. And here you just have a comparison with methionine PET. And you could see that, yeah, if you're using 2 different irradiation at 2 different frequencies here, at 3.5 ppm you have the AMIDE based signal, and at 2 ppm you have the amine signal. In a low-grade glioma, you have a nice separation between these two, while in a high-grade glioma, well, you'll see that actually, in this case, the amine signal is actually elevated a little bit everywhere, while actually in the low-grade glioma, it's only in the areas, which is the hot areas here around methionine. So it seems to be working. What about GLUCOCEST? Well, this was one of our first patients in lymphomas. So like, we decided, let's go outside of the brain. I ventured for once outside the brain. Well, that was not a good idea, I can tell you. In the first place, the neck is horrible. It's horrible because you have a windpipe here. And a patient definitely, generally, will actually swallow during an examination. So this will lead to differences in B0 and all sorts of problems in motion, because it's very complicated to actually align different necks, much more complicated than different heads, because the heads are kind of self-contained. The necks can be a bit different, but well, we got some pretty nice looking baseline images. Now the question is, we tried to do, at the time we were using a glucose clamps. That means we were just raising the blood glucose level and varying the speed at which we are infusing the glucose to maintain a same glucose level over time. And here are some of the results. So you have the data before and after, and yeah, there is something happening, but really, if I ask anyone, do you know where the tumor is? You wouldn't be able to tell. And actually, when you put some region of interest, either in the tumor or in the muscles here, over and over again, we couldn't actually see a signal that was actually significant. Now, this was a little bit disappointing, and I really wanted to show you that, because it's not actually that easy. And then we decided, well, maybe it's a bit too complicated. There's too many problems with B0 motion, and etc. Let's move back to the brain. And these are some of the first 2 results on patients from the Max Planck, in this case, where the whole the whole optimized sequence has been used. And you have here one patient, the first one with which is a high-grade glioma, and the second one with a lower grade. And you see that, well, you can see the tumor on T2, you can see the tumor on FLAIR, and you can also, with the dynamic contrast enhanced, look at the ring enhancing here area, while in this case it is not enhancing. And you do get a signal in the case of the high-grade glioma, but not in the case of the non-enhancing tumor. Now, you might actually want to know what the hell is this signal? Where does it come from? Well, I can tell you. Well, in the first place, it might or might not be representative of the gadolinium enhancement. They are part of the gadolinium enhancement ring that are actually high and part that are low. But there's also an increase in signal here in this necrotic part of the tumor. Now, is this interesting? In a leaky tumor, the glucose will leak into the necrotic part. And guess what? It's called necrotic part because it's dead. So if it's dead, it's not going to metabolize the glucose, and therefore it will accumulate there and stay high. So yeah, it's a good internal check that you can see it in a place where it doesn't disappear because it's been metabolized. But it doesn't actually tell you much about the metabolism of the tumor in this case. So the question is into how much clinical interest we will, clinically interesting information we will get there. I guess the jury is still out. And in case of the low-grade glioma, there was nothing to be detected really around there. So, conclusion. The take-home message of this talk, I hope it could show you, was like, you know, once you moved out of the chemistry lab and of Silvio Amy's wonderful world of chemistry and small animals, if you get into the clinic, things get a bit more difficult. It can be different. It can be difficult because you're using different field strengths. You don't have access to the same RF powers, and really, you're looking at different diseases because an animal model is not equivalent to a human with a tumor. It will never be. They are actually the tumor microenvironment and the interaction with the surrounding tissue is going to be completely different. And most of these animals, if you're using human cells, are immunodeficient, and this interaction between the cancer and the immune response is really what makes this unique characteristic and this very huge problem that we have to solve in human medicine. Now, the endogenous amide or amino CEST is readily available, and actually it was quite easy to translate it into the clinics, and the first actually FDA-approved clinical solution has been actually released by one of the vendors, which I'm not going to say here because I'm not allowed. But as for the GLUCOCEST, well, GLUCOCEST was very reproducible. There were many groups who did the same thing and could reproduce the results in a small animal. It turned out to be harder to detect at clinical field strength, and I'm not talking about 7T, but because the signal is small, there are problems with B0 and B1 inhomogeneities, and there are also problems of motion. So with that, I would just say maybe just that GLUCOCEST might actually be the killer application for human 7T scanners, but who knows? Thank you.